So we've talked about the land plants and how they fit in with other photosynthetic organisms in the world. We talked about cyanobacteria, the origin of photosynthesis. And today's topic kind of fills in some of the gaps we'd covered as far as organisms that can do photosynthesis and how they became photosynthetic in the first place. So the theme is photosynthetic eukaryotes. Land plants are eukaryotes, but so are a bunch of other organisms worldwide that can do photosynthesis. So pretty interesting how they're all connected. So just another refresher, you've seen this quite a few times. Just a reminder that organisms that are able to do photosynthesis are not all descended from the same common ancestor. So there's not one origin of photosynthesis in the groups that have them, have that ability. But there is a sort of a non-traditional way they're all connected that we'll see today. Okay, so if we think about eukaryotes doing photosynthesis, it also helps to think about where eukaryotes came from. So what is a eukaryote? You probably remember that from your early biology classes. The eukaryotes are defined as having a nucleus. So a membrane-bound nucleus is an organelle. In the eukaryotes, that's where the nuclear DNA is kept. So most of the genome contained in the nucleus, and that membrane is kind of important, enabling them to compartmentalize not only the nucleus, but other parts of the cell have membranes around them. So membrane-bound organelles are also a hallmark of the eukaryotes. So we'll see those play out a little bit as we talk about their importance to plants and photosynthetic organisms. So when we look at eukaryotes, eukaryotes also have a history of endosymbiosis. So maybe an obvious example or a well worked out example is the origin of the mitochondrion. So the mitochondrion actually descends from an ancient bacterial free-living organism that a eukaryotic cell took up into its body, into its cytoplasm, and sort of kept there as this, as it says, endosymbiosis, endosymbiotic relationship, the endo part inside of, and then symbiosis living together. So this is another organism living inside of the eukaryotic cell, and actually over many millions of years, becoming the, actually billions of years here, becoming the mitochondrion. So it was a free-living bacterial cell, and it became the mitochondrion over time, as this illustration shows. And this is important because mitochondria are central to just about every eukaryotic organism. So plants have mitochondria, our cells have mitochondria, and that's a little bit basically tied in with them being eukaryotes. So when we think about this endosymbiosis, there is a relationship between two organisms, or what actually started as two organisms. And for two organisms to live together and basically one not to outcompete or destroy the other one, there needs to be some kind of benefit to both. So there's a benefit to the mitochondrion and a benefit to the host cell. And maybe we'll talk about that with, why don't you think about that for a while? I don't want to give away all the answers here. Okay, so we talked a lot about eukaryotes and bacteria, and eukaryotes and bacteria are two of the three domains of life. At least that's how it stood for a while. So there was eukaryotes, bacteria, and this third domain called Archaea. Well, things are getting more complicated, as the slide tells you. And the Archaeans, this group of organisms that for many years got no study, hardly any study relative to the other organisms that we've talked about. And recently, people directing their attention at the Archaeans have found that they're not all necessarily descended from one clean Archaean ancestor. And that's what we really like to see. If we're going to name a group of organisms, we like to see them all coming from one origin and staying related to each other. So we've got a phylogenetic tree over here showing bacteria descended from a common ancestor. The eukaryotes down here descended from a common ancestor, but the Archaeans are coming out in 1, 2, 3 at least blocks of common ancestors. And what it basically shows you is that the eukaryotes are descending from sort of from within, they're evolving within the Archaeans. And actually very similar to the relationship we were to make an analogous relationship of the land plants to the green algae, is a very similar arrangement, but of course different groups of organisms. So where do the Archaeans stand now? Well, they're not their own group. They're still quite different from bacteria and different from eukaryotes. What it looks like is that the Archaeans are going to have to get divided down into sort of subgroups here. So don't worry about the names, of course, but this paper that I've highlighted here has them called Eukaryotes. Archaea is named for 4 letters abbreviating these different major groups of Archaeans. And this one's new to me this year, Asgard Archaea. So when you're hard up for names, you discover something new and you need to give it a name, why not reach into familiar literature? So they dug into Norse mythology. I don't know if you can read down here. This one is called Loki Archaeum, and there's a Heimdall Archaeota. So your favorite characters from the Thor franchise are now the names for some Archaean groups. And the phylogenetic tree is getting pretty darn complicated. So what is a eukaryote at this point? Well, we know the eukaryotes have a little bit of bacteria in them, and it looks like they've got basically an Archaean ancestor is what gave rise to the eukaryote. So this is our eukaryotic cell here, a little bit of a mix of bacteria becoming the mitochondrion and the rest of the body becoming, coming out of the Archaeans. And that was our original eukaryote, so billions of years ago still. Then what? So this is kind of new, maybe controversial. I think it's pretty exciting. It's actually spearheaded by somebody who's on faculty at UW-Madison, and he's on faculty in botany. So he kind of comes out of this botanical background and has allowed his mind to wander into the origins of life. And what he's been working on with evidence, not just a wild speculation here, but with some pretty good evidence, this idea that the eukaryotic cell didn't really start as it's usually thought of as this cell eating up other organisms and making them part of itself, but sort of starting from the center and enveloping things around it. So this is with the numbers progressing from left to right here, enveloping other organisms and kind of making use of them. And in that fashion, you can kind of see how this would also get you this interwoven membrane arrangement that we still see in the eukaryotes. So maybe a little bit more of a partnership extracellularly, even before the mitochondria became internal, might have been hanging around on the fringes of these ancient Archaean cells. And then ultimately they partnered up and became what we had as the eukaryotic cell. So this is supposing the origin of the mitochondrion, and we'll also talk about the origin of the chloroplast as we talk about these. So if we're going to have an organism that is eukaryotic and can do photosynthesis, then we have the eukaryotic starting organism. How does it learn to do photosynthesis? Well, here's where it ties all together. So we've seen eukaryotes that can do photosynthesis. We've seen bacteria that can do photosynthesis. And it turns out that to learn how to do photosynthesis as a eukaryotic cell, you can consume or take a photosynthetic bacterium into your body, as illustrated on the right side here, through that endosymbiosis process. Take it in, leave it inside of the cytoplasm, and it becomes this organelle that can do photosynthesis. So we had a cyanobacteria cell that can do photosynthesis. Now it's inside of a eukaryotic cell, still doing photosynthesis and giving the benefit, sugars, the energy, giving that benefit to the host cell, and that becomes the chloroplast. So that's the connection. The cyanobacteria originally could do photosynthesis, and really everybody who figured out photosynthesis after that, they were cheating and copying off of existing technology. So eukaryotic cell took up a bacterium, learned how to do photosynthesis. Didn't really learn, just sort of made somebody else part of its body and got to work. So the origin of the mitochondrion and the origin of the chloroplast are very similar to each other. There's lots of similarities if we look at the structures of these. The mitochondrion, for whatever reason, often gets painted in this orange color in the cell diagrams. The chloroplast, as we know, has a strong reason to be green. That's the chlorophyll in there. So we have two membranes, an inner membrane and an outer membrane. And the folding of the inner membrane is important to the process. We saw the inner membrane being important for segregating the charge during photosynthesis, positive charge inside, negative charge outside, and that was able to fuel the production of ATP in photosynthesis. So a little more detail on the chloroplast. There was a time when people had to figure out this idea that the chloroplast was originally its own bacterial cell. Now that's, I'd say, not really controversial at all. There's lots of good evidence for that, and I'll show you some pieces of evidence here. But sort of think about it as though maybe you didn't know about this. We'll walk through the evidence and you'll say, oh, neat, how cool. This is a thing that's behaving as though it's its own organism, but still living inside of plant cells. So chloroplasts are off on their own. They divide on their own. So when cells divide, they have to partition the chloroplasts that are inside of them between the two resulting cells. There are multiple chloroplasts per cell usually, so that's something that they would divide to increase their number, and then the cell division of the host cell sort of partitions them into different parts. Here's an illustration of a chloroplast in the process of dividing. They behave a little bit cellular-like. So they have their own DNA, they have their own genome, and their genome is a circle. So rather than the chromosomes we see typically in the nucleus of eukaryotes that are linear, right? They've got telomeres at the end, and they're just sort of long, single direction pieces of DNA. In the bacteria, and then also in chloroplasts and mitochondria, the genome is a circle. So this is an example chloroplast genome of a tobacco plant, and each of these boxes here is a different gene. So it's not a huge genome, it's not like organism-sized genome, but it has lots of genetic information for doing some of the processes that chloroplasts need to do. So it's got some evidence of having its own origin as its own cell. It's largely, in many ways, I guess I'll say, in many ways self-sufficient from the host plant, but not so self-sufficient that it could survive outside the host plant. Okay, so there is a chloroplast genome, and it's kind of large. It's not nearly large enough for that chloroplast to be able to do everything that a bacterial cell would need to do. So what we see here is a diagram of comparing living cyanobacteria. Those are these three on the right here, Seneca cystis, other cyanobacteria, comparing the sizes of their genomes. And this is in a number of genes, 7,000 genes, 3,000 genes. So that's kind of the amount required to live as a bacterial cell. But if we compare those to the genomes of the chloroplasts that are related to them, they share a common ancestor, chloroplast genomes max out at about 200 genes, so about 10 times less in the number of genes that they have relative to free living cells. So what has been lost? Well, over time, if you think about it, this free living cell has to get its own food, has to find its way through the environment, survive harsh conditions, gather light, deal with things that are trying to eat it, potentially eat or locate food sources in its environment. So there's a lot of concerns about surviving and gaining nutrients and things that single cells would need gene tools to be able to do. But if you then are a cell inside of another cell, you become a chloroplast, then the host cell can do a lot of that for you, can respond to the environment, can maintain safe internal conditions, can get nutrients, and so on and so on. So a lot of the genes that are essential for single-celled living organisms are no longer required for the chloroplast. So a lot of loss, and kind of a similar loss across all these different groups, probably related to their ancestors. So the ancestral origin of the chloroplast meant that a lot of genes were lost, genes that were probably redundant with the host cell. So the host cell was taking care of stuff, and the chloroplast genome could get a little bit reduced down to only what was left as being required. So what's left as far as function? You see these are the different colors here are photosynthesis, translation, and other. So proportionally, this green block is still pretty important. Imagine that. Genes required for photosynthesis get left in the chloroplast. So obviously, the cell, the eukaryotic cell that took up that chloroplast originally could not do photosynthesis, would not have the genetic tools for doing photosynthesis. So it needs to hold on to those because those are present in the chloroplast genome. Translation is kind of interesting. So the parts of the chloroplast that behave independently, it still does its own transcription, translation, gene expression. And so that all happens. It still needs the proteins required to get that done. And other, there's another category of other. So sort of the bare minimum that is required is what's left on the chloroplast. Kind of interesting. There's a little bit of a sign that says that, or evidence that tells us that the chloroplast has become really interdependent with the host plant. So the cell that holds on to this chloroplast, some of the genes that are still required for photosynthesis are actually no longer in the chloroplast genome. So they got moved to the nucleus. And this could be just a process of DNA moves around sometimes, random possibility. Could be that this sort of tied the chloroplast to its host cell. So it couldn't leave because it couldn't even do a complete set of photosynthesis without all the genes that some of them would be left in the nucleus. So the genes are present in the nucleus, in the chloroplast. Sometimes they have to be, genes have to be made into protein products outside of the chloroplast and then put into the chloroplast. And that's what's shown here. Lots more detail than you need to worry about, but just wanted to kind of highlight this. There are signals that can be put onto proteins that tell them to go into the chloroplast where they then would have a function. So lots of interesting molecular genetic, molecular biology connections here. More than we need to worry about. Okay, so it gets even more complicated. So the original eukaryotic photosynthetic cell would have been a eukaryote that then arranged with a cyanobacterium and then was able to do photosynthesis. And the evidence for that is that that basically happened once. And when that happened once, that gave rise to this group that we call the Archaeplastida. And Archaeplastida just means basically original or ancient, and plastida, plastid we're going to see is a word that relates to chloroplast. So the original cells to have chloroplasts were descended from this one event where a cell first was able to get a chloroplast. And the groups that descended from that, three big groups. The chlorophytes, chloro is green. So the root word for that, chloro, means green. Those are the green algae. Rhodophytes, the root for that is red, rhodophyte. And then this other group that we won't talk much about, but these are also in the Archaeplastida. They're called glaucocystophytes, or sometimes shortened to glaucophytes. So these are living organisms today that descend from the original endosymbiosis event that gave eukaryotes the power of photosynthesis. Whew. We also have evidence for what that cyanobacterium might have been. So this is a phylogenetic relationship fan among cyanobacteria. So all these ones with the dots at the tips, these are cyanobacteria. And the question might be, who is living today as a free-living cyanobacterium and most closely related to the original cyanobacterium that became a chloroplast? So we have a candidate here. This is the closest living organism that's been sampled so far that is related to the origin of glaucophytes, red algae, and green algae in plants. So hooray for that. So this is also, I guess, highlights another neat feature of that chloroplast genome, is that the genes present in the chloroplast can be used to tell us what that chloroplast is genetically most similar to. So it has some really ancient genes in there, things for the ribosome and so on. Some ancient, highly conserved gene regions that can tell you who the closest living relative of chloroplasts are. Okay, so algae, I've kind of mentioned this term before, this idea that the blue-green algae are not algae. What about the rest of algae? Are they all descended from the same group? Not quite. So there's not a single common ancestor for things that we call algae. This is kind of a problematic term when it comes to classifying. So we don't like to use these terms or classify groups anymore that don't have a single common ancestor. So we've seen that green algae and red algae, they could have a single common ancestor. And they do, as far as we can tell. But keep in mind also, of course, the land plants are basically still in the group of green algae. So we have to kind of carve them out separate from the land plants. And then we have this group of the brown algae. And they are one we haven't talked about yet from an evolutionary perspective. This is going to be a group that was not descended from that Archaeplastida single chloroplast origin. Dun, dun, dun. All right, but a little bit about algae here. We have different kinds of algae: red algae, brown algae, green algae. These are probably worth noting as much as anything because they are have members that are visible to the naked eye. So these are some of our macroalgae. So within red algae, I just highlight some maybe familiar ones here. Some of the algae that we have in the red algae group are used as the organism that gave this agar for our petri dish plates. So that kind of jello, gelatinous solution that sets up your nutrient agar. You can grow bacterial cells, could even grow algae cells on there. Sort of a scientific jello, and that sort of the binding agent comes from red algae. One that you might have seen in your ingredient labels, carrageenan, is a red algae product from this one called chondrus. So red algae gives us that. I think this might be from, well, I suppose that's almond milk or maybe almond ice cream. Carrageenan is a thickener. So it's a little bit like what we talk about with agar, something that sets up and makes molecules bind together a little bit more. This can be used in food products to give you some thickness to your ice cream, your milkshakes, your almond milks, and so on. And then also the wrapper on sushi rolls is a red algae. So basically an intact leaf, what we call a leaf of a red algae, dried out, wrapped around sushi rolls and holds them together. Brown algae, we'll see a few of these. So diatoms are brown algae, or basically belong to the brown algae group, as are some of these rather large, really macro algae called kelp, for example. This is a brown alga. Looks a lot like you would expect a flowering plant to look. So it has things we call leaves and things that kind of look like roots and parts that look like stems. But it really doesn't have an ancestor shared with the land plants that have what we think of more as proper leaves and stems and so on. So just a convergence on the useful form of a multicellular organism to have. Okay, so let's find our brown algae on here. Let's see if we can. Maybe we can't. They're going to be in this heteroconts group. And not labeled as brown algae, so that's a problem. All right, why don't I start a new video for us? This is getting a little long. Stop.